Hi everyone, welcome back to Mathematics. Today I'm going to talk about the bonds and reactions which are present in DNA and RNA. In my previous videos, I have already talked about the structures of DNA and RNA. So let's look at the bonds and reactions which are present in DNA and RNA today. So firstly, I'm going to talk about the hydrogen bond, which is present in DNA helix. And then I'm going to talk about the phosphodiester bond and the condensation reaction. These two bonds and reactions are present both in DNA and RNA. So let's look at the hydrogen bond first. So hydrogen bond means the attraction between a hydrogen atom and an electronegative atom. So the electronegative atom examples are oxygen, fluorine, and nitrogen. So these molecules will possess a polar negative charge and the hydrogen atom will have a polar positive charge, and thus there will be an attraction between these two charge, and that is why it is also known as a dipole-dipole attraction. So there are two types of hydrogen bond present in one DNA helix, and these two types are one type is like there are two hydrogen bonds present between adenine and thymine, and the other type is there are three hydrogen bonds present between cytosine and guanine. So let's look at the hydrogen bonds in a bit more details. So first I'm going to talk about the bonds between the adenine and thymine. So here we can see that the nitrogen and oxygen atom, they are electronegative molecules and they have a negative charge on them, whereas the hydrogen molecules in both cases have a positive polar charge, and that is why there is an attraction between these two positive and negative charges, and hence there are two hydrogen bonds present between adenine and thymine. Now let's talk about the cytosine and guanine. Here we can see that this oxygen atom and this nitrogen and this oxygen, they all are electronegative atoms because they have negative charge on them, whereas there are three hydrogens which have polar positive charge, and then these three hydrogens have created hydrogen bonds with these three electronegative atoms. So there are three hydrogen bonds present between the cytosine and guanine. So by forming the hydrogen bonds, the DNA helix is formed because these two strands are attached by this hydrogen bonding formation between the adenine and thymine and the cytosine and guanine. So we can say that the DNA helix, like the strands, are bound to each other by forming hydrogen bond with each other like this. So there will be two hydrogen bonds between adenine and thymine and three hydrogen bonds between cytosine and guanine, and thus these two anti-parallel strands are attached to each other by forming hydrogen bonds between the nitrogenous bases present in each of the DNA strands in order to form the DNA helix. So now let's talk about the phosphodiester bond that is present both in DNA and RNA. So this phosphodiester bond is basically the bond between the phosphate molecule and the sugars that are present in DNA and RNA. So this phosphodiester bond is also known as the backbone of the strand, as it helps to attach two nucleotides together. So this phosphodiester bond basically forms a linkage between two sugars in RNA and DNA. So it basically binds to the third carbon atom in one sugar and the fifth carbon atom in another sugar. So let's look at how this phosphodiester bond is formed. Okay, so this is basically one single-stranded RNA structure formation where we can see that in one nucleotide, in the fifth carbon position of the sugar, there is already one phosphate molecule present. So let's look at how this phosphate molecule will bind to another sugar in the third carbon position. So we can see that there is one hydroxyl group present in the third carbon position of this sugar of the nucleotide. So when this hydroxyl group will come across the hydroxyl group from this phosphate molecule, this will go through a condensation reaction where one water molecule will be lost, and hence there will be a phosphodiester bond formation, and that is how this nucleotide will be bound to this nucleotide in the formation of the phosphodiester bond by losing one water molecule in the third carbon position. Okay, so let's talk about the another type of condensation reaction that is present both in DNA and RNA. So we know that the nitrogenous base joins the sugar in the first carbon position. So how does this happen? So here we can see that there is one hydrogen atom present in the adenine structure, and here is one hydroxyl group present in the sugar structure. So when these two come across, there will be another condensation reaction, and we know that in the condensation reaction we will lose one water molecule, and thus there will be a bond formation between the nitrogen and the nitrogen from the nitrogenous base and the carbon from the sugar, and then this nitrogenous base will get attached to the sugar in order to form nucleotide structures in both DNA and RNA. And that is how the nitrogenous bases are basically attached to the first carbon position in the sugar. Thank you for watching this video with me. I hope you guys have enjoyed the video, and I will try to come up with some more concepts, and I will try to explain them as much as possible. I will try to make these concepts clear. Till then, keep loving science and stay safe.